Hello friends, in last video we shall discuss about the mitosis cell division. Means there are three types of the cell division: amitosis, mitosis and meiosis. We are going to discuss about the meiosis stage. Now this next cell division, meiosis, and this cell division, it is called the reductional cell division. Now this meiosis, it takes place in the reproductive cells, and this meiosis is firstly observed or firstly coined by J.B. Farmer in 1905. Now this meiosis, takes place only in a reproductive cell during the formation of daughter cells. Now this mitotic and meiotic, these are the type of the cell division. In mitosis cell division, it is a equational cell division, and meiosis, it is a reductional cell division. The reductional means it is reducing the number of chromosomes. They get the haploid number of chromosomes. And now this division, meiosis, this cell division, the number of chromosomes are reduced to half, and now it is called the reductional cell division. Now this meiosis, it is a special type of the cells, and this cell division, it is necessary for sexual reproduction. This cell division is necessary for sexual reproduction process. This sexual reproduction means some eukaryotes, this cell well organized nucleus. Now this eukaryotic type of the cells, this type of the reproduction process is occurring, means this sexually reproducing organism, means this meiosis is occurring. They can begin takes place the division of cell. The diploid cells, they can containing the two copies of chromosomes are found in the meiosis cell division. Now this meiosis cell division, there are some stages, the meiosis first and meiosis second. These are the type of the cell division. In meiosis first and meiosis second. These are the two stages of meiosis cell division, like the meiosis first and meiosis second. Now we shall discuss about the first meiosis first stage. Now this first meiosis first stage involves the prophase first, metaphase first, anaphase first, and telophase first. These four phases are including meiosis first, and same meiosis second, the prophase second, metaphase second, anaphase second, and telophase second. These are the cell division. It is occurring in the meiosis cell type. Now the first type, meiosis first, include the first phase, prophase first. Now this prophase first, it is the longest phase of the meiosis, and this stage, meiosis first stage, this meiosis, it is the reductional cell division, and meiosis second, it is the equational cell division. Now the first type of meiosis first includes the first phase, prophase first, and this prophase includes the subphases. First phase is liptotin. Second phase, zygotine. Third phase, pachytine. And fourth phase, diplotin. And fifth phase, dikinesis. These are the 5 phases of prophase first. Now the first phase, these are the interphase, liptotin, prophase first include the subphases like the liptotin, zygotine, pachytine, diplotin and dikinesis. These are the 5 subphases are included in the prophase first. Now this first meiotic division, the diploid cells, means this 2n structure, they can convert into the haploid cells, means the reductional cell division. In the meiosis first, it is the reductional cell division. The diploid cell is converted to the haploid cells. The chromosome number is also reduced to half for each cell, and this cell, it is called the heterolytic division. This phase is called the heterolytic cell division. Now this prophase first. Starting about the prophase first, it is the longest phase, long duration, and it is a significant phase which is particularly it is occurring in the meiosis. In these phases, the first liptotin. Now the first phase is the liptotin. It is interphase. This is a resting phase. Now this interphase means already discussed about the cell cycle. This interphase is the resting periods of cell division, and in this phase it is occurring the this G1 phase, S phase and G2 phase. And now this interphase, they can next phase, M phase, means meiotic and mitotic cell division. Now this first phase, it is called the liptotin. It is the first begun this first prophase, and now this liptotin inside of the cell, the nucleus is enlarged. In this structure of this liptotin cell, this nucleus is an enlarged structure, and in this phase the chromosomes are elongated structure arises, and these chromosomes, they are distinct, but it is a long thread like bodies are formed, and it is known as a chromatid. These chromatids are formed in this liptotin phase, and these chromosomes are long elongated thread like bodies are formed in the first phase, prophase first, liptotin. Now this liptotin phase, the centromeres, it is a duplicate and migrates toward the opposite poles. Now this is the first phase, liptotin. The next prophase first is second phase, zygotine. Now this zygotine phase, it is a next phase of prophase, and it is called the zygotine. Zygotin phase, it is includes the homologous chromosomes. Now this homologous chromosome, they can take place the formation of synapses. Now this synapses means the homologous chromosomes, they are appear or they are present in the pairings. They are formed the pairings. Means the synapses process is start in the zygotine phase. Now this zygotine means the pairing of homologous chromosomes. Means one chromosome is a maternal and another is a paternal chromosome, and these two homologous chromosome, similar chromosome, it is forming the pairings. Now these chromosomes are forming the pairing in the zygotine phase. Now the next phase is the pachytine. this Pachitin phase. Now this phase zygote pairing of homologous chromosomes takes place, and later this phase formation of 4 strands means this tetrad formation is occurring in the Pachitin. In these chromosomes they get pairs, but these 2 pairs are grouply present in the Pachitin stage, and these 4 chromosomes are appearing in the groups, and these 4 homologous chromosomes are present in the one position, and now this group of these 4 chromosomes means the formation of 4 strands of chromosomes. It is called the tetrad. With the Pachitin stage, this Pachitin stage, the homologous pairing means 2 similar chromatids there appears visible and form the 4 visible chromatid. Means the homologous chromosomes of each pair, they become each other and they are present in this Pachitin stage. Now the next phase is a diplotin. Now these 4 pairs of chromosomes are found in this phase. In that phase, diplotin phase, this chiasmata formation takes place in the also in the Pachitin, and diplotin phase, the distinct cross-like structure means this phase, the chiasmata formation takes place in the diplotin phase, and this chiasmata is already formed in the Pachitin stage, and later they are clearly visible. This chiasmata is clearly visible in the diplotin stage. The next process for this diplotin, the exchange the chromatid means crossing over takes place in the diplotin phase. These sister chromatids are present in groups, but in this phase, diplotin phase, the exchange of chromatid means the maternal and paternal chromosome exchange their chromatid, means crossing over is occurring in the diplotin stage. Means it is called the Dikinesis, means it involves the disappearance of the subdermal complex. Now this process, diplotin phase, DNA recombination takes place in this phase, and chromatids contain 2 shortened structure. This chromatid, they get shortened structure. In this phase, it is the longest thread-like structure of chromosome, but in this diplotin phase, these chromosomes exchange the chromatid, but their shortened structure is formed in the diplotin phase. Now the next phase is a dikinesis. Dikinesis phase. In this phase, it is a last phase of prophase first, end of the prophase first. In this phase means the terminalization. In this phase, the terminalization means the chiasmata is formed and they can move to belongs to chromosome. They can form the centromere. This middle portion, centromere, is formed, and toward the end of the chromosomes means the displacement of the chiasmata in dikinesis phase. Now this dikinesis means terminalization. This phase, it is the nucleolus and nuclear membrane, they get disappearing. In this phase, the nuclear membrane, nucleolus, they are disappearing in the last phase of prophase first, dikinesis. This phase, they can disappearing, and the spindle fibers, they can started appearing the spindle fibers in this phase, starting the spindle fibers in cytoplasm in the dikinesis phase. These are the 5 stages of meiosis first, prophase first. Now this prophase first include these 5 phases. The first one, liptotin, zygotin, pachytin, diplotin and dikinesis. Now the second phase, the metaphase second. It is the prophase first. Next phase of meiosis is metaphase first. Metaphase first. Now this metaphase first, this phase, now this last phase of the prophase dikinesis phase, in this phase the Chi's meta formation is occurring, and now this metaphase form the spindle fibers. The metaphase occurring, the spindle fibers are formed, and now these chromosomes, they get appear in the middle portion, means they are present in the middle portion. Meta means middle. These chromosomes are arises in this region, means middle region, and now this middle means the equatorial region. These chromosomes are arises, they can present in the middle portion in metaphase first. In this meiosis first, metaphase first, the chromosomes, they get appear in the middle portion. This metaphase first. Now the next phase, anaphase first. The meiosis first, anaphase first. Now during this phase, in this phase, the homologous chromosomes, they are full away from each other, and they can carrying or they can toward the opposite pole. This middle portion of chromosome, they get separated in anaphase. They can separate toward the 2 opposite poles, and this phase is called the anaphase. This anaphase, formation of spindle fibers, and this middle portion of chromosome, they get separated in the anaphase. They get present in the 2 opposite pole in anaphase. This is a disjunction. Now these 2 sister chromatids of each other, they do not separate in the meiosis first, and now this meiosis first, it is called the reductional cell division. Means the chromosomes or sister chromatids are separated. They can equally they can separated or reduce the half number of the chromosomes in the anaphase first. Now the next phase of meiosis, telophase first. Now this is a last phase of meiosis, and this phase is called the telophase first. Now this telophase first, this one cell, they get divided into 2 daughter cells are formed, but the formation of 2 cells, that time the middle cell wall, means the cell wall, cell plate formation is occurring in between these 2 cells, and later these 2 cells, they get separated. Now the formation of 2 identical cells in the meiosis first. Now this haploid number of chromosomes are after reaching a respective poles become uncoiled and elongated, and this last phase, the nuclear membrane, they get appearing, the chromosomes, they get long elongated structure, and these 2 daughter cells are arises in the last phase of telophase first. And last phase is the cytokinesis. Now this cytokinesis, after the karyokinesis process, the cytokinesis is occurring and 2 haploid cells are formed. This cell plate formation is takes place, later they get separated toward the opposite, these 2 cells are formed, and that time the cytokinesis process is occurring, means cytoplasm divided into 2 cells, and these 2 daughter cells passes through the short resting phase, interphase. Now in same change of the telophase. Now it is a last phase of the meiosis first. Now in this video we shall learn about the this meiosis first cell division, and this meiosis first present the prophase first, metaphase first, anaphase first, and telophase first, and cytokinesis. And this prophase first, it is the longest phase of meiosis first.